Great. Great to see all of you today. First of all, we would like to begin by acknowledging that this seminar is happening on the traditional, ancestral, and unceded territory of the Musqueam people. And for those online, you may be joining from traditional lands across British Columbia, Canada, and the world. And we would like to acknowledge the owners and caretakers of those lands as well. And with that, I would like to introduce today's speaker, who is the first speaker of our Fish 500 seminar series. I suppose we don't need to have any more detailed introductions of Curtis. Curtis is a professor here, also a university distinguished scholar in the IOF, but he also appointed by many different departments, EOAS, Microbiology and Immunology, and Botany. And he's also the Associate Dean of Research in the Faculty of Science. And Curtis is a world-leading expert in ocean virus, and particularly on this diversity, evolutions, and function in ecosystems. And with that, he holds many different titles and awards. He is a fellow of the Royal Society and the American Academy of Microbiology. He recently appointed as the Order of Calendar, as well as many other distinguished awards. I won't list it here, but it's a lot. It's a long list. I will take up all his time. So with that, we all look forward to hearing more about ocean virus today from Curtis. I pass it to you. Thank you.
Thanks very much, William, and thanks everyone for being here. As I say, I'm usually a roamer, and so being tied to the podium, I'll do my best here so I don't walk away. I'm just going to put a timer on here so I have some idea how long I'm taking. So, okay, so yeah, so today I'm going to have a little bit of a walkthrough, maybe some of the things that we're doing, and for those of you that don't think about microbes in the context of oceans and fisheries, then I hope to kind of introduce the concept that it's really difficult to actually understand the oceans and the life within it unless we also consider the microbial organisms that are also there. And so I'm going to give a few vignettes and maybe a few general comments, and we'll go from there. So first of all, I just want to say everything that I talk about and everything that's done in the lab is not done by me. It's done by the fantastic people that we work with. This is a recent picture of the group, but not recent enough. Some people we've already had some turnover since this photo. So, so all the work I'll be talking about today was certainly done by, led by others. And I also want to acknowledge again, as William did, that we're on the ancestral unceded territory of the Musqueam people, but also I think it's really important because when we sort of think about it in terms of the context of knowledge and learning, that the only city that I'm aware of that was actually named after a First Nations leader, a chief, is Seattle. And so this quote, which is attributed to Chief Seattle, is that the earth does not belong to man, man belongs to the earth. This we know, all things are connected like the blood, unless unites one family, all things are connected. And so I think that's really appropriate in the context of this, because again, when we think about the oceans, we think about ecosystems, we think about processes, everything is connected. And often we have a very narrow view of what we work on. We drill down and we become specialists in terms of what we work on, but we sometimes lose the connection of all the other things that are important. So where we work, again, is at the University of British Columbia is where we're based, on the unceded ancestral and traditional lands of the Coast Salish people and the Musqueam. But my lab is in the Biodiversity Research Center, so just across the way. And but we also work all around the world. At least it's taken us so many places, from the high Arctic to the Antarctic to hydrothermal vents, and even occasionally we go into deep mines. And so this is the Crystal Caves in Mexico, where we went looking for life in extreme environments. So we've had the privilege of kind of going all around the world. And so what we do is we study what I'll say the virosphere. But the virosphere is that part of the earth where viruses exist and that are affected by viruses, which essentially is everywhere in the earth. And so when I say we study the biosphere, we're actually studying the earth in a sense, right? Because everywhere there is life on earth, there are viruses and other microbes as well. And so what we generally do in a broad sense, and I'll talk about some of these things, but not everything today. I'll touch on a few of these things. We do discovery. So I'll focus on that a fair bit today. So what that means is we go in and we discover essentially new life forms, new groups of viruses and other microbes. We've worked with NASA about how do we go and find life on other planets when we don't know what we're looking for example. So we have a kind of long history in that. We look at diversity and evolution of the microbes and the viruses. So try and understand the extent of their variation in the natural environment, but also their deep evolution. Where did they come from? What are they connected to? And we're very much interested in their role in natural systems. And so that goes from mortality to global biogeochemical cycles, right? And so I certainly won't have time to touch on all of these things today. But one thing I really try and emphasize in my talks, excuse me, is that if it weren't for viruses, we would not exist. And people don't often think about that, but we have a long, deep evolution with viruses. And so one example is that, for example, the placenta in mammals contains a number of proteins which were co-opted from viruses, right? So viruses which infected us got stuck in our DNA and became repurposed, right? So there would be no placenta if it wasn't for viruses. There would be no mammals. We can look at our nervous system, and some of the main proteins in our nervous system are all also co-opted viral proteins, structural proteins from viruses that over a long evolutionary time of our deep ancestors, this isn't recent, but our very deep ancestors, our single-celled eukaryote ancestors co-opted these proteins and repurposed them. And ultimately, they continued to be repurposed, right, through evolutionary time. So I think it's really important that this long, deep evolution, it's a very long, deep evolution. I'll talk about that a little bit more. And then I'll also, I won't dwell on this, but hopefully by the end of this, you'll realize that without viruses, we could not survive. And the reason why is because viruses are so intricately connected to global biogeochemical cycles, right, and processes. And so we are absolutely dependent on the processes that are driven by viral host interactions. So, and we can go back and think about the origin of life. So if we go back and think about that, well, viruses have been around for a very, very long time. If we look at the formation of the Earth about 4.6 billion years ago, something like that, 4.7 billion years ago, the hydrosphere developed incredibly quickly, right? So the planet became an aqueous planet within a few 100 million years, which is remarkable, right? Considering it was just a ball of molten rock, right? And so we had a hydrosphere very, very quickly. And there's evidence of the first life at about 4.2 billion years ago. And that's based on looking at carbon, that as far as we know, carbon compounds, which are only made by living organisms, they're only understood to be made by living organisms. So if that's the case, life started very, very quickly, right? And so if we think about that development of life, we see things like stromatolites, which are aged back to about 3.6, 3.7 billion years ago. And the thought is that those were formed by microbial processes. And so almost certainly viruses were there from the very, very beginning. And we know, for example, that the major groups of viruses were there before the cyanobacteria split off from the other alpha proteobacteria, because we see the same groups of viruses, the evolutionary same groups of viruses infecting all the cyanobacteria affect all the bacteria, other bacteria, the non. So that means that the viruses were likely there well before that split. And so again, so if we look along this long lineage, what I want to emphasize is for at least a couple billion years, life on Earth was only prokaryotes and the viruses which infected them. And so if we think about that evolution, how deep that evolution is, so it's viruses and their prokaryotic hosts that drove biogeochemical cycling, right? The only predators in the system were viruses, right? Until some bacteria evolved the ability to tackle and destroy other bacteria. But for the most part, the ecosystem for a couple of billion years was only prokaryotes and the viruses which infected them. And so I think it's really important to keep that in context when you think about how co-evolved these systems are and how long they've been interacting together. So viruses and life are certainly not a new thing. And in fact, the first eukaryotes only showed up about 1.6 billion years ago, the first pretty solid evidence for eukaryotes. So we see some evidence of some multicellular organisms. We have fossil evidence, but the first skeletonization, or the first organisms that seem to be able to deposit carbonates, was only about 570 million years ago. So I mean, that seems like a long time, but it's not a long time over the evolutionary of life on Earth, right? So when we look at where most of the diversity resides, the multicellular organisms, although we're very phenotypically diverse, biochemically, we're pretty limited, actually. So in terms of what we do. So the first couple of billion years, point here, life was aquatic. Life was exclusively microbial. And viruses were likely there from the very beginning, and evidence of skeletonization and things like that is only about 570 million years ago, right? So relatively recent in the evolution of life. So lots of people don't think about viruses, and I know we all are kind of familiar with viruses, but I'm just going to really quickly say viruses are ultra microscopic infectious agents containing nucleic acid, a genome, and usually a protein capsid. They replicate, they're obligately dependent on replication in other organisms. Is that a characteristic only of viruses? No. And so I think this is really important to recognize that there's lots of obligate parasites which are obligately dependent in terms of replicating on organisms. They cannot replicate on themselves. So that's not just a feature of viruses. It's a feature of an ultimate parasite. And are viruses alive? Now, I would argue yes, but I don't have time to go into that here, but I'm happy to talk about why I think viruses are alive, if anybody wants to ask me afterwards. How big is a virus? A virus is really small, okay? So, and I was giving a talk in Toronto for a philanthropic organization to a bunch of donors, and they asked, could I give a talk, an after dinner talk, right? And I thought, well, gee, I mean, these are all smart people and everything, but they're all lay people in terms of science. So I thought I should think about this. And I hadn't really thought about how to describe how big a virus is. So it's about 100 nanometers, which seems pretty small. So the average North American is about 1.7 meters tall. And I was in Toronto, so I was at the CN Tower, and I went up to the CN Tower, and I thought, I know how I can explain it. I could say, if I scaled myself to the size of the CN Tower, and I scaled a virus to the same amount, how big would the virus be, right? I thought that would be a good way to kind of communicate this. And I hadn't thought through this very deeply. And so the virus would still be invisible. Okay, so that's not a very useful simile or something, or to try and describe it. So that's not very helpful. So I thought, well, what if I did it the other way around? And I said, well, okay, let's take something we can see, the pinhead, and let's scale a virus to the size of a pinhead and scale myself to the same size. How tall would I be? Okay. And so I take a virus, scale it to the size of a pinhead, and I scale myself to the same amount. I would be between actually 25 and 125 kilometers tall, right? So the point being, viruses are incredibly small, right? And yet they have such consequent effects on processes that are happening. And so again, it's interesting. I look back, I gave a short talk in the IOF actually 10 years ago. It was like a little mini talk, and these are the same slides, right? So these ones right here. So because I still need to get this across, so many of you weren't here 10 years ago, right? So if we look at the number of viruses in the ocean, for example, or we take a little bit of seawater and we stain it with nucleic dye, which stains nucleic acids, right? That's what all these little fluorescent dots are. And so we took water and we did that. Each of these little dots are actually a virus particle. And each of these little bigger dots are a bacterium. And that big thing in the middle is a protist. Okay, this is kind of what life on Earth looks like. So if we were an alien sending a probe to Earth, chances are we'd hit the ocean. It's about 71% of the Earth's surface. We're very unlikely to hit a whale or a fish or anything like that. And if we retrieved our sample back, this is what we would see. And we would conclude that life on Earth is microbial. And And that's largely true. So how abundant are these things in the ocean? So on average, there's about 10 million virus particles in a milliliter of coastal seawater, and about, bacteria, and about 50 million virus particles per milliliter of seawater. And what does that mean? That means every time you go swimming in a lake or in the ocean, you swallow as many viruses as there are people in North America, right? Just by the water you take in your mouth. So these numbers are big, and so they're kind of hard to think about, but that's how abundant how abundant they are in the oceans. And if we were to look at that in the context of stars in the universe, for example, there's about 3 times 10 to the 23 stars in the universe. This person got the paper in Nature because they realized we had underestimated the number of stars by 3-fold, right? And so, but if we compare that to viruses in the ocean, there's about 10 to the 30th viruses, or about 10 million-fold more viruses in the ocean than there are stars in the universe. And if you stretch them end to end, they would go about 10 to the 7th light years, which is further than the nearest 60 galaxies. And I think that's important because I just said how small they are, right? But that's how abundant they are just in the oceans, right? And so I think that's important when you think about it, because those viruses, there's about Avogadro's number of infections per second, right? About 10 to the 24th infections per second in the ocean. And every time there's an infection, there's an opportunity to laterally transfer nucleic acids, have major impacts on evolution. So when colleagues say, oh, but that would hardly ever happen. Well, if it's happening 10 to the 24 times a second, we may not be able to see it, but it could be happening all the time, right? And so I think that's something to keep in mind, that life is continually experimenting. And when you think about how long we've co-evolved with viruses and the number of infections that are going on continuously, there's so many opportunities for new properties of life to emerge, right? And so again, just to put it in context, so if we look at viruses by numbers, they're by far the dominant nucleic acid containing or life form in the oceans, right? And I'll talk about this in a little bit, but these viruses then are responsible for killing about 20% of the living material by weight, not by numbers, by weight in the ocean every day. Okay, so there's an incredible amount of mortality coming along, and that has consequence effects on lots of different things. And they're not only in the sea. Some work that we did a number of years ago with Isabelle Richet, and she had a bunch of stations on top of the Sierra Nevadas in Spain. And she was collecting things in collectors. I said, why don't we look at the viruses which are falling out of the atmosphere? Because her stations were above the planetary boundary layer. And that means in the area of the Earth where wind is not impeded by surface friction, right? So where the jet stream is, where wind can freely circulate on the Earth. And so we did that just because I had data, and I don't have time to go into it, which suggested that, well, actually, if you're a microbiologist, you know everything is everywhere, but it's kind of hard to explain sometimes, right? So why do we find viruses, the same viruses, like every environment that we looked in, when clearly the host could not be in all of those environments? And so we collected these things, and we had some atmospheric modeling to say where the winds were coming from. And the amazing thing is that this is just falling out, where this is passive collection, about 10 to the 9th viruses per meter squared per day falling from the atmosphere in the most pristine environment on Earth, right? So once you're above the planetary boundary layer, you are freely circulating around the Earth, which is why we see everything everywhere when we look at viruses. Okay, the dispersal is enormous. And so I mentioned that a lot of the stuff that we do is discovery. And so when I talk about discovery, most of what we do is discovery of microbes and viruses, and specifically which infect other microbes, like microeukaryotes or bacteria, right? Or different kinds of phytoplankton, for example. So we've found all kinds of new families of viruses and things like that over the years. But what I really want to spend a little bit of time today is 3 things that maybe are a little bit more easy to connect to, right? So examples of where we've been looking at viruses which infect what I would call charismatic megafauna, right? But Maybe some of you don't think an oyster is a charismatic megafauna, but for me it is, right? So I like to eat oysters, and I think they're beautiful. So I'm going to two stories. So the first one is by Gideon Mordecai, who's I think a research associate. I think his position is at the Institute of Ocean Fisheries, who was a postdoc in my lab when we started this. Some work by Kevin Zong, who's a research associate in my lab, and Melanie Prentice, who was a research associate in the lab and just moved on to be a Hackeye scientist. So I want to talk about these three stories in a little bit of depth. And so again, because these are examples of discovery. And so one of the questions are, we know that many stocks of Pacific salmon have been declining dramatically over many years. And the cause of those declines are not really obvious, but we ask the question, is it possible that pathogens might be involved in this? And so this is in collaboration with scientists at DFO. And what we did is what actually the DFO scientist did, Christy Miller, was go out and collect dead and dying salmon, which were dead and dying of no known causes. So she has a big array in which she can look at all pathogens that are known to infect salmon. And so if these things were dead and dying, but there were no known pathogens that infected them, so then what we did is do deep sequencing. So we go into those and collect these fish, essentially do what's metatranscriptomic sequencing, or see what things are being expressed, what kinds of genes are there, and look for novel virus genomes. And what was really remarkable was the number of viruses that we found which were completely unknown in salmon. And some of these appeared to be clearly associated with disease. And so you can see on the list here all of these new viruses. So everything that we looked at was new, essentially in salmon. And we were able to demonstrate or show infectivity in many of these cases, right? And so what was really cool is, again, because of our collaborations with Christy Miller's group at DFO, they've been sampling salmon smolts for years. And so what we could do, and sequencing them, so we could go back and interrogate 6,000 salmon smolts and look for the presence of the viruses in these different smolts, in these different fish. And so what we see here is a heat map. Essentially, black means the virus was not detected. So these are different kinds of viruses. These just abbreviations here. So they're very, very diverse in terms of the types of viruses that they are. And you can see in some cases, they're really, really abundant and really hot. Other cases, you can see that there seem to be inland. So we can see that the distribution and occurrence of these viruses is very different, which means the epidemiology is very different, right? So in other words, what's happening is very different among the different types of viruses, how they're interacting with their hosts. And I think it's very, very complicated, because with a virus, you have to ask the question, does it kill it? Does it make it sick? Well, in some cases, we can be pretty sure. And in one case, we found the virus actually came from Norway. So it was clearly associated with Atlantic salmon originally, and it was now circulating in wild Pacific salmon. And it looked like it caused disease in wild Pacific salmon, but not really in Atlantic salmon, right? And so because it's a host range expansion. So, so you can start asking these kinds of questions, but they're complicated, because sometimes you see lots of viruses and then just a little ways away you don't see any, but you don't know, is that because the fish died? Or is it because their immune system was able to fight the infection and sweep the virus away from the fish? So those kinds of questions become very, very difficult. And those are the kinds of questions that scientists at DFO are very interested in trying to address. So again, so here's an example of the kinds of things that you can do by this kind of de novo discovery, because we know very, very little about the organisms which infect, the viruses and the organisms that they infect. So if you look at almost any marine organism, even something as important as salmon and as well studied as salmon, we know nothing about the pathogens almost which infect them, right? Another story is a program that we've been involved with in many years called Sustainable Shellfish Aquaculture. We were trying to understand what the causative agents of mass mortality of oysters. And this is a global problem. In some cases, pathogens are known, but in many of the cases, and especially in British Columbia especially, the causes of these mass mortality events are unknown, and they're extreme. Like when they happen, almost every oyster dies on a farm. And it's absolutely devastating to the growers, which tend to be fairly small operations. And so what we did as part of this is we went on an expedition along the coast of British Columbia, and all these little dots are places that we stopped and collected oysters. And then we came in where these two little red dots are, we came in to get water in Deep Bay or Bain Sound, right, which is where most of the oyster farms are, and they were just pulling up trays of oysters and literally everything was dead and dying. They were pulling them up to harvest them, right? And a week before everything was fine, and they pulled them up and like every oyster is either dead or dying. And so we kind of jumped on that and so said, can we try and figure out what this pathogen might be? And so this was Kevin Zong who kind of led the analysis of this. And so we brought the oysters, we sampled them, we brought them back, did the same thing, except rather than a salmon, now we've got oysters here. We did the metatranscriptomic sequencing, and we tried to look for novel virus genomes. And what Kevin was able to pull out was a really interesting virus. It's an RNA virus with the largest genome ever seen, right? And it was unknown in bivalves. This group of viruses had not been seen in bivalves before. And it was clearly associated with mortality. So all of those other samples that we collected from oysters all around, we never saw evidence of this virus. We only saw this virus associated with this mass mortality event. And so from an evolution perspective, it was really interesting in terms of the type of virus it is and the characteristics of the virus, but it's also very interesting from a natural history story as well, because we still don't know for sure that the virus caused the disease. All that we know is that the virus was strongly associated with the mortality event. And so when Kevin looked out at metagenomic or metatranscriptomic databases around the world, he found that this virus was found in Pacific oysters in Asia and in Europe, right? So everywhere that the Pacific oysters were grown, at least in the Northern Hemisphere, we found the virus associated with it. And what we also found, which was really interesting, here's a molecular phylogeny of the viruses. So the virus is actually a distant relative of coronaviruses, right? So it falls in the same order. So of the coronaviridae, which is SARS-CoV-2, right? So we're familiar with that. So it's in the same broad group. But if you look at it, here's all the viruses. And so you see the red ones are Canada and China and Europe, France, and you can see that they're almost all identical to each other. So what that suggests to me is that the virus spread very quickly, right? Very likely through translocation of seed, which is what oyster farmers do. They buy their seed from all over the world, right? And so it's very likely that this is how the virus moved around. So, and then the third vignette that I'd like to mention is not a virus, but we thought it was a virus when we started, right? And so this has happened many times to us. We've got lots of papers of completely unknown bacterial pathogens. We isolated one bacterial pathogen which has no complete biochemical pathways, none. All it does is encode transporters because it's an obligate parasite. So it looked exactly like a virus, couldn't grow, couldn't do anything. And then when we sequenced the genome, it's highly reduced genome, which was completely streamlined for parasitism. That's all it could do, right? So there's many cases where we think we've got a virus, and then we pursue it more deeply, and we end up with a bacterium. And so this case, this was a group that was pulled together by Alyssa Gemmon, who's here, but was led by a research associate in the group, Melanie Prentice. And Amy Chan is a really important player here because she isolated the pathogen, which actually is able to cause this sea star wasting disease. So again, the experimental setup wasn't terribly difficult, but where it started was taking the coelomic fluid out of the sea stars, which had signs of wasting, which I think was something that others had not done. And then Amy was able to isolate, when we looked at it from a sequence analysis, Melanie looked at it from a sequence analysis, it was just basically a single bacterial taxon. And we could grow it. So Amy was able to isolate it, grow it in culture, and then we took it back and we reintroduced it back into the sea stars again, and it invariably caused wasting, right? And so there's a lot of work that was done in this, many years of work done in collaboration with folks at the Maristone Lab in Washington and USGA Lab. And I think what we did is we conclusively demonstrated that this bacterium can cause wasting, right? So that doesn't mean all wasting is caused by this bacterium, but we have evidence that it's probably important because by going out and mining data from the past, when the pandemic initially started, we were also able to find the bacterium Vibrio pectinicida associated with sea stars that were wasting in the field as well from before our discovery of this. So it's really, really interesting. And so when we started, we've sequenced the entire genome of the bacterium, and we find some really interesting genes that are likely responsible for toxicity, right? We see some what we call pathogenicity islands, right? So again, so here's another example. Again, people have been trying to figure out what the pathogen was for more than a decade. And there's been numerous papers published. Sometimes, certainly in the long term, have not had much support, and in other cases just didn't make a lot of sense, right? But I think in this case, we really took a very experimental approach, and through luck and the people that were working on it, we were successful. So I think the point here is that I really want to make is that there's a strong interaction even when we look at sort of populations and population dynamics. And so if we're interested in macrofauna, you know, whether it's, you know, seagrasses, sea stars, or sea slugs, right? There's going to be a strong association with microbes which infect it and cause disease. If you look at humans, there's 140 at least viruses alone which make us sick and cause disease. That's just viruses. That's not including the bacterial pathogens, of which there's hundreds, and also the protistan pathogens like Giardia and malaria and all these other things as well. So there's no reason to think that the same isn't true for every organism, right? So the fact that we find a pathogen doesn't mean it's the only pathogen ever, right? But what is interesting is what causes an explosion or what causes a sudden change in the dynamics, right, between a pathogen and a host interaction. And then I want to just kind of, how's my time? I just wanted to kind of finish up and talk a little bit about other ways that viruses affect us on a global scale, which I think is really, really relevant, not only to oceans, but also to fisheries and to ourselves. So in the oceans, about 95% of the living material by weight, not by numbers, is stuff you can't see, right? So if you could take a giant balance, 95% of it, of the weight is stuff you can't. We, you know, our sensory abilities only see, you know, those wonderful things that we see in the ocean, but most of the stuff that's happening, we can't see. And so to give you a reason why it's so important, those microbes are responsible for about half the oxygen production on Earth, right? So it's very, very dynamic, right? Processes are really, really fast. And viruses, as I mentioned, kill about 20% of this living material by weight every day, right? And so you can imagine that not only shapes a species, right? Because of the way viruses are special because they tend to be highly specific. So with Kevin's work, I didn't mention in detail, but when he looked at all of the, at these viruses infecting Pacific oysters, they were only found in Pacific oysters. He looked at, I think, 8 different species or something like that, but this virus wasn't found in any other species of oysters, right? So viral pathogens tend to be highly specific, right? That's why a norovirus, which infects a seal, is not going to give us norovirus disease, right? Which is, you know, because unfortunately, when we get norovirus from an oyster, eating a raw oyster, which has been contaminated by, that's human sewage, right? So that's the problem, right? So, but the thing that's really interesting is viruses are that specific, and that's why they're so amazing at being able to create diversity and shape ecosystems, because they replicate fast when populations get large. And we know that, everybody knows that, right? So if somebody's sick, you don't want to be in a room with someone sick, because if you've got a high density of people, likely people are going to get sick, right? And so that's the way viruses regulate populations. It's called kill the winner. They knock down who's ever growing fastest, right? And they allow things which have other traits which allow them to succeed, right? Under different conditions. So viruses, if we didn't have viral infection, the diversity on Earth guaranteed would be an awful lot less diverse, right? So, and the other thing that we don't think about is because of that mortality, they also drive biogeochemical cycles. So we did experiments where we took viruses out of seawater and primary production stopped. And that's because the system is dependent on viral infection, right, for recycling nutrients, right? So again, so that's other ways that these things kind of interact. And so what I wanted to mention here was just a couple of other examples, which not my work, but I think is really interesting. So this is Land's End in the UK, right? The southern England, that's the English Channel, and you see these white blooms. And so these white blooms are caused by a coccolithophorid, right? So a little phytoplankton, which is really globally important, right? This little species of phytoplankton. But the reason it's white is because the blooms have been lysed by a virus, right? So this population is you see these massive blooms and these massive lytic events. And so what's the consequence of that? Right? The cliffs of Dover, right? So this has been going on for millennia, right? For millions of years. And so the accumulation of coccoliths on the bottom of the English Channel actually ultimately built up the cliffs of Dover, right? So these kind of interactions between viruses and their phytoplankton hosts in this case, this is sort of a very visual representation about, yeah, so there are kind of global consequences going on. The other thing, when we kind of think about it, we published a paper many, many years ago. This was a postdoc in my lab at this time, Stephen Wilhelm, who's now like a chaired professor in a university in the US. And we came up with this idea of what we call the viral shunt, and that's how the viruses cause lysis of microbes, and that material is recycled through the system in what we call particulate dissolved organic matter, right? So again, this is a chemical process, and that organic material is taken up by those million bacteria per mil, right, which is in seawater, right, and respired and recycled, and the nutrients come out, right? And so then that material, that process goes on and on. So in fact, that's why primary production stops when you take the viruses out, because you stop the lysis and you stop the recycling of the nutrients, right? But another consequence of that, which we hadn't really thought about at the time, right, but is really, really important, is in terms of on a global scale, this is really important. So if you think about like CO2 and where the giant reservoirs are, so the biggest reservoir is fossil fuels and things like that, right? So there's teratons of carbon tied up in fossil fuels. In the atmosphere, this is about 835 gigatons right now, right? So that's the total amount of CO2 in the atmosphere. It's going up by about 3 gigatons a year. And then in the oceans, though, it's 39 teratons, right, of inorganic carbon in CO2, but it's bicarbonate. And so the point that I want to make here is that where most of the carbon is actually dissolved in the ocean. And so if you look at kind of where carbon comes from, if you look at deforestation, fossil fuels, that's a couple gigatons a year, right? And so we have production, right? 5 gigatons per year, so deforestation, fossil fuels. But venting out of the ocean is 90 gigatons a year, right? So 10 times more than all of the other carbon being vented, right? That's caused by humans. But we're lucky that 93 gigatons per year is going into the ocean, right? So it's being sucked up. What's the consequence of that? Ocean acidification, right? So we're lucky it's actually going into the ocean in some sense, but there are definitely consequent effects. And those 3 gigatons per year end up being sinking out into the deep ocean. And so this is what we call the biological pump. And so where do viruses go in here? Because they cause lysis, right? And they cause a lot of cycling, right? So in the dissolved organic carbon pool. And so if you think about that process, if you think about the microbes that are being lysed, like bacteria and phytoplankton and things like that, right? So most of that biomass in the ocean, a lot of the carbon in those microbes is pretty recalcitrant, right? Like cell wall material, peptidoglycans, cellulose, right? All kinds of things that are kind of hard to break down, right? And why is that important? Is because if we look at the dissolved organic carbon concentrations in the ocean, we have a bunch of different pools that we categorize as oceanographers. And one of them, the largest pool by far, we call recalcitrant dissolved organic carbon. So that carbon, if you just add it up, is more than all the carbon in the atmosphere. and its average age is 6,000 years old. So why is that? That means it's not respired, right? It's biologically inert. It's really sequestered, right? So it's sequestered. It's not having climate consequences, right? It's not being broken down by microbes on any scale that we can measure, right? So it's a way to kind of capture carbon, right? Or it's the way that the ecosystem captures carbon over the long period of time. And so why are viruses important in that? Because they essentially fuel what we call the microbial carbon pump. So what happens is viruses are responsible for lysing 20% of the biomass a day. All the stuff that's used easily, right, like sugars and proteins and stuff like that, the bacteria that are there break that down right away. Things that they really need, like dissolved inorganic nitrogen and dissolved inorganic phosphorus, like phosphate, nitrate, ammonium, all of the iron, all of these things which are absolutely required for life are taken up immediately. And then you have more photosynthesis, right? Because photosynthetic organisms make carbon. They're not dependent on carbon, right? They just need that other stuff, nitrogen, phosphorus, iron, everything else. And they got lots of light because they're in the surface of the ocean, and they're not short of water, right? So they're plants. They got everything they need to grow. So primary productivity goes fast, right? Because of all this recycling. But what gets left behind? That stuff that doesn't get broken down, right? That recalcitrant dissolved organic carbon. And so again, if we think about consequences on sort of, this is one of the other things that we look at, from a global biogeochemical standpoint, viruses play an incredibly important role in capturing carbon naturally. And so there are people thinking about ideas about if we could accelerate the pace at which primary productivity generates and viral lysis occurs, which is actually pretty easy to accelerate, then you could actually increase the rate at which carbon is captured. I'm not suggesting that we geoengineer the planet. I'm not a big fan of that. But these are processes, natural processes that are occurring that it's really important that we're aware of, and how the different things, how climate, et cetera, kind of changes the rate at which these processes occur. Okay, so hopefully, if I kind of think about this is like oceans and fisheries, right? So if we think about, I gave you some vignettes, which I think tied in kind of nicely to the kind of why viruses and microbes interact strongly with the macrofauna that we're dependent on, the economically and ecologically critically important organisms. I don't know how you can understand population dynamics of macroorganisms if you don't consider the microbes. And most people don't consider the microbes at all, right? And yet they're probably the most important thing driving population dynamics, right? In many, many cases. And the second part that I want to just I gave you one little story about just how these microbial processes really drive the planet and how tightly that we're associated with them. So I just want to thank you for being here today. And I certainly thank everybody that past members and current members of my group, which allow us to do these really interesting things. Thank you.
Thank you, Curtis. We have a bit of time for some questions, either online and or in the room. Go ahead, Sarah. And Curtis, for the people online, if you just summarize the question because they can't hear people in the
Gotcha.
Thank you. That was fascinating. Your top 10 years ago, all of that's living in virus thing at Harvey was one of my favorite. Yeah, but my question is, I was struck by the graph or the map that showed the oyster infections and how they were concentrated in the farm oysters, like the wild oysters, healthy. And I was just wondering if that's basically a density reason, or do wild oysters, are they resilient to that disease for other reasons, like genetic diversity, or should we be worried about our wild oyster population?
Right. So great question. So the question is about why we only saw the viruses, this Pacific oyster nidovirus associated with the farmed oysters, and we didn't see any evidence in the wild or the naturalized oysters at all. And so that is a really great question, and we don't know the answer, but it begs a lot of interesting questions. For one thing is the growers, they get their seed from elsewhere, right? And they, in many cases, historically have imported it from other countries, from Chile, from France, from the UK, from the USA, from all over. And we also produce oyster seed in British Columbia as well, but for a while we were having a lot of trouble. So people were actually sourcing their seed from elsewhere. And so the question is, because we only saw it in these farmed oysters, we have some suspicions that the seed came from elsewhere. Was the virus imported with the seed? We don't know, right? We still don't know that the virus actually caused a mortality event, but we're pretty sure it did based on the kind of virus it is and how tightly associated it was with that mortality event. So I don't know. It could spread to the beach oysters. It could have just come in into seed, right? And so I always tell the growers this is actually a good news story because now we actually know what we're looking for. So we can actually interrogate seed if it's coming in, or we can find out if this pathogen might be associated with it. But we don't know, right? But the interesting thing is, you know, when you look kind of everywhere, the only thing that everybody looks at are farmed oysters, right? Nobody looks at wild oysters, right? Basically as well. So all the data that we have are also from farmed oysters as far as I know, right? And so where we find it in other oysters in other parts of the world, but there are also no reports of mass mortality there. Doesn't mean it wasn't occurring. I mean, growers are pretty hesitant to talk about problems.
So, sorry, there's a related question online, Curtis, which I think you answered at the end. The case that you uncovered in Canada, are there similar cases elsewhere in the world for farmed oysters? Or was this sort of like a new thing, you know, where the mortality was only occurring or the virus was only found in the mortality events?
Okay, I assume that you could probably hear that online, given that David's standing right here. So the question is, again, we don't know, but we do know that unexplained mass mortality is a global problem, right? It's happening all the time. And where other pathogens have been found, they've been found in many places, right? So like oysters, there's a herpes virus, which causes disease in oysters, right? And so that's devastating. Once they found it in one place, they could find it in other places. So I think now we can start to develop the tools to try and answer that question.
More questions? In the back. Nope, yep.
Right, great question. How do you decide what tissues to sample when you're looking for something that you don't know what you're looking for? And you know, it's in oysters we tried to grab the hemolymph and we tried to grab the mantle tissue. Fortunately, we found the virus in both. You know, typically because sometimes we lost the mantle tissue or sometimes we lost the hemolymph, but it seemed like the virus occurred, you know, in both places, which perhaps isn't surprising. In salmon, again, what we ended up taking were gill tissue primarily because it's easy to collect and it's also associated with the blood. And it's interesting, some of the viruses that we found that were really abundant in the gill tissue were also respiratory viruses, which had never been seen in fish before, right? But were associated with the respiratory system in salmon. So it's hard when you're dealing with a big organism, right? And we were involved, for instance, in what's killed all the belugas in the aquarium. And so, you know, we were getting tissues from all kinds of organs and all kinds of things and doing deep sequencing on it. And we never found a pathogen, but we did get the first complete genome for a beluga whale, right? So that was our contribution, right? So, but we were unable to find what caused the demise of it. So, you know, you just don't know. And it's a hard question because you don't know what you're looking for. And I mean, we're fortunate now that we do have methodologies, some of these deep sequencing methodologies. They remain expensive. They always say sequencing is so cheap. The problem is that the quantum unit of sequencing now is about $30,000. It's true, you get a lot of sequencing, right? You get huge amounts of sequencing, but it's hard to do that experiment where you say, I'm going to blow $20,000 and I hope it works, right? So that's kind of the key, but this deep sequencing allows us to interrogate samples that we never could before. So a lot of it's best guess. Like sometimes you'll see, if you see pathology, you'll target those particular areas, or if you think that something's going on. Yeah.
Amanda. Oh, okay. Is your student? Oh, Colin, you don't count as a student. Go ahead. Yep, Vicky.
Like about a decade ago. And I know you mentioned that one of the big problems was like you thought it was virus and then this video really reduced bacteria, streamlined for parasitism. What would you say was like the most time-consuming thing? Was that, or was it like there's just a billion bacteria because of viruses affecting us? So here's the one concept.
So again, great question. Like, I don't know how I can kind of summarize it, but how do we go about, kind of going from a virus to a bacterium? And so one thing is this is not a streamlined bacterium. This is kind of the opposite. This is a bacterium which just can do about anything, right? So it's very, very plastic. It can pull in genes from other bacteria, pathogenicity genes typically, right? So cholera would be an example of this. So cholera causes disease when it's infected by two different viruses, because the viruses actually encode the genes which cause cholera to be toxic, for example. So the original hypothesis was based on kind of the experimental approach that they used, where they did, again, lots of sequencing on diseased sea stars. And they sequenced the heck out of them, and they found evidence for a virus which seemed to be associated with that sea star. Turns out, yeah, but like a lot of things, it wasn't only associated with diseased sea stars. And that's not necessarily wrong, because just like all of us are full of strep, but we can be killed by strep too, right? So it just because you have something associated with you doesn't mean that it's necessarily going to cause disease. And so in this particular one, it was actually we did a bunch of experimental things, mainly kind of size fractionated sort of things, and it seemed to exclude almost all viruses except very weird viruses, which I wouldn't expect to see in a sea star, right? So that kind of gave us the hook, and then we said, okay, let's see if it's bacterial. It looked like it could be bacterial. And so we just, it's pretty easy at that point to go and say we're going to just look for bacteria. There's different markers that you can use that you can get data pretty quickly. And it turned out, wow, it's all bacteria, right? And so we didn't have any evidence that it was viral. And so again, it's sort of kind of fortunate in a sense. And so I think it's easy to say that the other experiments weren't very good. I sort of think like that because we took a very methodological approach, very, very experimental, because that's kind of that's what I like to do, and which the other scientists in the group like to do as well, which is we're going to do testable hypotheses and do experiments that are conclusive in their outcome. So we'll do this and we'll do that. And if this happens, that means this is the case. And if that's the case, we'll do the next experiment, right? And so we did the full circle and then came back, isolated, purified the pathogen, reintroduced it to the sea stars, caused disease in all cases, right? So that's called Koch's postulates, right? Of doing the full circle, right? So I think it's hard to know. It's easy to always second guess, be an armchair quarterback and say, oh, what was the problem? But I think maybe the fact that we went after hemolymph was one of the things that the others hadn't done, because if you're doing microbiome work looking for disease, surface swabs are the worst thing you can do, or grinding up an organism to look at what's there, because everything is there, right? That's the problem, is like everything is there, right? And so it's really hard to try, and you can do all kinds of principal coordinates analysis and all these sorts of things and trying to say, oh, this is associated with disease, but it's always really messy, right?
One really quick question. Okay.
I'm really interested in the relationship between fishing pressure and viruses. And I'm wondering if you would imagine that populations under pressure might end up being more vulnerable to viruses in such a way that fisheries are actually creating a compensatory impact by the remaining fish that weren't fished now being more vulnerable to viruses.
Wow, so that's a tough one to answer, right? Whether fishing pressure could in fact induce disease. But the one thing I think we can say sort of in a short answer is that many things are multi-stressor. So just like with ourselves, if we have, if we're fighting, well, take an organism like oysters, for example. If they're fighting high PCO2 levels, they're fighting temperature, they're fighting, they're fighting maybe a Vibrio infection, things like that. So their immune system is compromised, it's already overworked. So if something that stresses an organism definitely can make it more susceptible to infection. But one of the really difficult things as well is that things, again, have evolved over millennia. And so it's like, oh, this thing's infected, I can jump in there and I can take over, right? And so and this is why HIV took forever to try and figure out, because when you get HIV, you get all these other pathogens associated with you as well. And everybody says, oh, it's this pathogen. It's not HIV. HIV came in because this other pathogen was there, right? And so these kind of codependencies and co-occurrences are really, really hard to sort out. So ultimately, just like we did with the sea star, if it's possible, which is very hard to do with humans, you can't take HIV and inject it into humans and see if you can make them sick. But with invertebrates, we can still do that, right? And so that's the thing where you have to be able to close that loop at all possible.
Okay. Thank you. I know there's many more questions. Curtis, are you going to be able to join us?
Yeah, I'll be there.
Okay.
Yeah.
Down the hall. For those of you who came in, first of all, I'd like to join me, please, in thanking Curtis. So great start for the seminar series.